So today I'm going to talk to you guys about our EMSL capability called TerraForms. Many of you have heard about this, and I think a lot of people, a lot of you present today have also seen many of these capabilities. And we think that most of these capabilities that we have grouped under TerraForms are really useful for studying soil processes. Specifically because soils are really heterogeneous and opaque and difficult to image. So we came up with these reduced complexity platforms that help us answer specific hypotheses, which is really difficult to tease apart in soils. And so today I'm going to talk to you about several different reduced complexity platforms, and together we call them TerraForms. These are tunable environments for exploring soil processes. So for specifically, these TerraForms platforms, they decrease and control complexity in soils. Like, for example, if you have a soil core and there's so many things inside of a soil core, like you will have roots, microbes, minerals, and combined together, this is a very complex environment. And so what we do is inside of these platforms, we can mimic parts of these specific elements of soil, and we decrease and control this complexity while maintaining the heterogeneity and thereby increase measurement resolution. And so some of the things that we can reproduce in these platforms are soil aggregate morphology, soil porosity, mineralogy. We can grow plant roots and study rhizosphere processes, microbes. We can add microbes in there, organic matter, and nutrients. And in these platforms, we can then characterize using several different types of chemical imaging capabilities that are present in EMSL, SSRL, and other places. And we develop workflow so that we can transfer samples across labs, which is really important for imaging these platforms. So one thing to mention is while all of these platforms are reduced in complexity, therefore we must use them together with like conventional techniques so that we maintain a check of like the processes that we are measuring, if they're correct. Just to tell you how we fabricate these platforms, we have different platforms that we can create using photolithography in the clean room. We can also make them using 3D printing or laser etching. And so just as an example, we can start with a glass cover slip. We make these platforms conductive, just so that they are compatible with some of our mass spec techniques. We build the actual habitat on top of that and then cover it with a membrane or a cover so that like it's an enclosed environment, enclosed space where these incubations can take place. And these are a couple of platform examples that we have. All of these are compatible with various types of biochemical assays, in situ chemical imaging. So you can take these platforms into the mass spec or perhaps X-ray fluorescence, some of the examples that I'll be showing you later on. And they're also compatible with omics, different types of bulk omics techniques, and optical microscopy. You can track microbial growth and plant growth inside all of these platforms. So that's the non-destructive part. So as I mentioned, we have developed several different workflows, and some of this you're gonna hear about these workflows in the next talk by Jocelyn. But most of these chemical imaging techniques that we currently have, these are destructive. And so we have workflows where we first do the incubation, track growth using optical microscopy, and also in the end, we are able to collect biomass through processes such as lyophilization, so as to end the experiment at that point of time, and then send them to various types of characterization, like mass spec or to a different lab even. And so those different types of workflow has been developed, where we start with incubation, sample prep, and so we can remove biomass from these platforms, separate the biomass while maintaining the structural integrity of the biomass, and then we can actually look at the biomass separate from the minerals or soil-like aggregates that's present in the platforms itself. And so in this way, we also have developed workflows to transfer these samples across different labs, and then, if required, bring them back here for further characterization. And so I'm going to describe some of the platforms that we have currently here at EMSL. We have platforms called pore scale micromodels. These are our smaller platforms, which are used only for microbial incubations, and some other work that we have done with users, such as looking at organic matter mineral interactions. That's one of our current projects. And so this is an example platform where we grew microbes inside of this particular micromodel. These platforms mimics soil pores. For example, in this particular platform, you can see these dark circles, and there is the clear space in between where the fungal hyphae is growing. So these, the platform that you see here is a very homogeneous patterned platform, but we can also fabricate these representing the soil porosity. I'll talk about that in next couple of slides. And so in this particular platform, we were tracking fungal growth, where the fungi is growing from one carbon hotspot C1 to C2, bridging these two hotspots through this soil-like pores. And then we characterized the fungi, the fungal hyphae, using different types of mass spectrometry techniques. We characterized using TOF-SIMS, looked at what the fungi was translocating in the fungal hyphae, for example, potassium. And then we did mass spec on the chip itself, where we looked at presence of different organic acids made by the fungi. And then further, we characterized this particular sample by sending it to SSRL, where we looked at the potassium chemistry of within the fungal hyphae. For example, as the fungi was making different types of organic acid and uptaking potassium, so we looked at, we found that there were different types of potassium chemistries inside of the hyphae, and we were able to detect that at SSRL. So this is an example where we transferred these samples across different labs, and we developed this workflow where we are able to separate biomass from the habitat itself to specifically look at to characterize the biomass that is growing inside of these habitats. Another of our platforms is the rhizochip, where we can grow plant root. And here we can also do plant root microbe interactions. So for example, root interaction with bacteria, root interactions with fungi. This is something that we have done for several of our user projects. So here's an image of different types of plants that we have grown. We can grow plants up to a month. In these, after that, the root architecture becomes too big for the chip to support. And then these are also compatible with mass spec. And an example here where we have analyzed the plant root using mass spectrometry imaging, multi-mass spectrometry imaging, looking at different types of phytohormones, amino acid derivatives, and other metabolites exuded by the plant root. So in this way, we can look at not only plant root exudate, but when plant root and the microbe interacts, what changes occurs in the exudates. And then we have other platforms called the subtep microbial and plant platforms. These are 3D printed platforms and was inspired from a user, EMSL user. And so in these platforms, we are able to grow plants and microbes. These are very high throughput. There are two platforms. One is the culture plate, where we can culture plant and microbe, as you can see here on the far left side. And then the blue plate, which goes underneath the culture plate, is where we collect the exudates, and it gets analyzed by mass spectrometry. And since these are 3D printed, these can be printed in any conformation as the experiment requires. So some examples of plant growth, we can grow them we can grow plants on agar or soils in these particular platforms, collect metabolites on the bottom plate.
Sorry about that.
And so another example of imaging the root architecture once the growth is completed. Then I'll move to the next platform. It's our bioprinting platform, where we can create these 3D printed soil-like aggregates. In this, we can print using this hydrogel. And in these small aggregates, so this is an example of how the printing takes place. We can print these with cells like microbial cells, bacteria cells, or fungal spores, and then they grow inside of these aggregates, and we can add minerals or organics inside, so that we can look at interactions inside of this 3D printed environment. So here are some examples of imaging inside of these 3D printed synthetic soil aggregates. We can do imaging of these microbes and by staining them inside of these aggregates. Here are some more examples of that. And then the other thing is these are also compatible with several different types of multi-omics characterization. DNA and RNA extractions can be done from these aggregates. So then we can compare these with if you have some kind of soil experiment as well. Okay, and then so this is our last capability within the TerraForms capability that I'm going to describe. It's our newest capability called Pore2Chip. Using this, you can create your own micromodels or rhizochip. So we start with the soil core. Users submit the soil cores here, and then perform XCT, where we get a bunch of parameters, for example, pore spaces, pore throats, pore diameters, and then we also get the pore connectivity. And so using this data, we can create TerraForms design. It could be, these are very scalable, so we can create these as a really small chip, which is only for microbial incubations, depending on the experimental needs. However, these also can be scaled up so that if you wanted to make a bigger chip for growth of a plant, that could be done as well. And so the advantage is that you could optimize these according to the pore spaces that you want. And depending on the pore space, it depends on the resolution that you want. We can make them using laser etching, which is really fast. Per chip probably will take around 3 minutes. But however, if you want a resolution which is less than 15 microns, so if you want something like 2 microns or something, we will make these using photolithography, which takes time. So depending on resolution, it could be a really fast process or not. But it gives you the capability of mimicking parts of a soil core, and specifically the pore connectivity, which I think is really essential when studying nutrient transports, any kind of nutrient, carbon, or even other elemental transport inside of these pore spaces. So as an example, we have we have printed cores where we looked at disturbances such as freeze-thaw disturbance, where users have submitted soil cores from Alaska soil, where there is like freeze-thaw disturbance, where the core has shown that there is a loss in pore connectivity versus an undisturbed core where there is not much loss in the, where there is no loss in the pore connectivity. And we can compare those as well using this particular technique. Okay, and so just as to wrap up, I think right now I'm gonna just show you this slide here where we have done a lot of integration using these reduced complexity platform. Tamas showed this slide as well. So using these new capability port to chip, we can actually do one more connection there with the soil pore connectivity, and then connect them to these different chemical imaging techniques, which shows a really nice story of integration. And then finally, I'd like to acknowledge this is a lot of people who have helped in development of these this whole capability. There are users who really tested some of our platforms before we actually launched them, so that we can really test out problems. And then finally, I would also like to thank the PNL communications team who made this workshop possible.